Welcome to our first lecture on the anatomy and enzymology of fat absorption. On the right, you can see a beautiful image of a micelle. We will go into detail about the micelle shortly. The micelle is an important part of fat digestion that holds fatty acids and monoglycerides. Bile is a non-enzymatic solution created from the hepatocytes of the liver. Bile is made up of bile salts, bile pigments, and cholesterol. We will focus on bile salts for fat digestion. Bile salts help to facilitate enzymatic fat digestion and are made from bile acids conjugated with amino acids. Bile salts act similarly to dish soap. They help to make fats soluble during digestion, just like dish soap helps to remove all the food remains from your plate. Bile is secreted into the gallbladder via the hepatic ducts and it is concentrated there. During meals that have fats, the gallbladder will send bile into the duodenum via the common bile duct and aid in fat digestion. The bile salts are then reabsorbed by cells in the ileum and sent back into circulation. Some bile salts make their way into the colon, and they are converted back to bile acids by colonic bacteria. Bile salts return to the liver via the hepatic portal vein, which you can see in this diagram here. And for every meal, bile salts will recirculate approximately 2 to 5 times to process all of the fat. Most digestion occurs in the small intestine. Intestinal, hepatic, and pancreatic enzymes are secreted to aid in digestion. The pancreas also secretes lipase, colipase, and pancreatic phospholipase to help aid in enzymatic fat digestion, and we will go into these in a moment. And here you can see a beautiful picture of a pancreas where it's secreting lipase, colipase, and phospholipase into the small intestine. In the American diet, fats and other related molecules, including triglycerides, cholesterol, phospholipids, long-chain fatty acids, and fat-soluble vitamins. Triglycerides are the primary source of lipids. Most lipids are not very water-soluble, as we have learned about previously. Chyme from the stomach has large fat droplets that are broken down by bile salts so that they can have a larger surface area for enzymes to work on them. Bile salts are amphipathic. This means that they have a water-loving and a water-hating side. The water-loving, otherwise known as the hydrophilic side, interfaces with the aqueous chyme, and the water-hating hydrophobic side interfaces with the lipid. This creates a lipid-soluble droplet. Lipases are enzymes that are involved in the next step of enzymatic fat digestion. Lipases remove 2 fatty acids from each triglyceride molecule. This creates 1 monoglyceride and 2 fatty acids that are held within the droplet. Now, because lipase itself cannot dig into the bile salts, another cofactor called colipase is necessary. And what colipase does is it moves some of the bile salts to the side, and this allows lipase to work on the fats inside the droplet. So the two working together is how lipase enzymes work. And so lastly, pancreatic phospholipase is another enzyme secreted that digests phospholipids, as the name suggests. And finally, cholesterol can be absorbed as is, and it does not need to be broken down before absorption. The next step after lipids are broken down by lipase, the monoglyceride and the 2 fatty acids resulting from the degradation of a triglyceride are stored inside of a micelle. As we have seen in the first slide of this lecture. A micelle is a small, round, circular disc that houses the products of fat digestion. They consist of a hydrophobic tail region that interfaces with a monoglyceride and two fatty acids, and a hydrophilic head that interfaces with water. The micelle will then proceed towards the cells of the small intestine. The micelle allows fatty acids and the monoglycerides to become water soluble by being inside this region here where my mouse is at. I hope that you can see that. And so that it can pass through an aqueous environment without interacting with the water, and can continue to its target. So once the micelle reaches the cell of the small intestine, the fatty acids and monoglycerides that are stored inside these micelles are then released and absorbed by the small intestine via simple diffusion. These can just cross the plasma membrane, no problem, just down their concentration gradient. However, cholesterol is a slightly different story. Cholesterol is transported into the cell via a transporter called NPC1L1, and cholesterol will meet with the fatty acids and monoglycerides once they're both inside the cell. This diagram over here illustrates how fatty acids and monoglycerides are packaged into micelles, and then they are released and absorbed into the membrane of the small intestine cell, and then the processes that occur following absorption. So we can see over here that we have the large fatty acids and monoglycerides, then they're covered in bile salts, and then what's missing here is there's a lipase and colipase come in here and break down the fatty acids into one monoglyceride and two fatty or break down the fat droplets into one monoglyceride and two fatty acids packaged into micelles, which then release those products, and fatty acids and monoglycerides cross the membrane by simple diffusion, and cholesterol will enter the cell via a transporter. So once the monoglycerides and fatty acids are inside the cell, they'll go to the smooth endoplasmic reticulum, where they recombine into triglycerides again. The cholesterol that enters the cell will combine with the triglycerides and the proteins to create something that's called a chylomicron. So this is a large droplet that is then subsequently packaged by the Golgi apparatus, and these are packaged into smaller secretory vesicles, and these are bound for exocytosis into the rest of the body. So these secretory chylomicrons that are created after the Golgi apparatus are still a little bit too big to go right into the capillaries and enter the bloodstream that way. So the chylomicrons are absorbed into the lacteals of the lymphatic system and enter the venous system just before the right atrium, the heart. So that's how they re-enter the circulation of the body. But however, there's still some fatty acids that are not assembled into chylomicrons, and some of these shorter fatty acids that are 10 carbons or fewer in length, those ones can enter the capillary system just that way and re-enter circulation. So that covers how we go from fat droplets into fat degradation and absorption, and how they're recycled and absorbed into the body. And yeah, we'll talk with the next lecture.